In our last lecture, we have discussed about the importance of protein to acquire their native structure, and in that video, I have briefly discussed about one example of quality control mechanism that is exist in the cell, in human cell. So I have not discussed that quality control that example in detail in that video. So I will do that thing in this video. So this we will understand the quality control mechanism in the cell in the subcompartment suborganal level. In the suborganal, that is endoplasmic reticulum. In endoplasmic reticulum, there is a quality control mechanism that is occurred at the time of post translation modification. So what happens is that when the protein is translating inside the ER lumen, the inside portion of the endoplasmic reticulum is called ER lumen, endoplasmic reticulum lumen. And in the, as we understand that there are 20 amino acids that in different sequence form a chain of amino acid, or we can also call it as polypeptide chain. So when this Glycan structure is shown over here in this, let's not go in too much detail how these Glycan structures are formed. Let's understand that this two square thing represent N-acetyl glucosamine, and this red circles represent mannose, and these three black circle represent glucose. So this Glycan sequence can bind to a chain, can bind to a sequence that is NXT. What is N is asparagine. X could be any amino acid except proline, and S or T, and third amino acid should be S or T. S is serine or T is threonine. So when this Glycan binds with this chain of amino acid or polypeptide chain, from now onwards we will call it as a polypeptide chain. So this will now this Glycan will now is recognized by another enzyme that is called glucosidase 1. So what glucosidase is 1? What it will do is it will cut off this first glucose. It will cut off this first glucose. This glucosidase 1 will cut off the first glucose, and then glucosidase 2. Glucosidase 2 will cut off the second glucose. And now this single glucose, that is monoglucosylated Glycan, is now recognized by either calnexin or calreticulum. Calnexin and calreticulum are called chaperones, as it helps proteins to acquire their native form. Usually this places in the cell, usually the place in the cell is too much crowded, so protein might get a non-native interaction from its neighboring protein. In order to avoid that, what happens is this now the polypeptide chain with this Glycan structure is now recognized by calnexin and also by calreticulum. So whenever they come in contact with calnexin or calreticulum, the calnexin calreticulum cover this polypeptide chain and allow them an isolated environment so that they can form their native structure. And after this glucosidase 2 will again cut off this third glucose. Now it is non-glucosylated Glycan. Now this structure is no longer recognized by calnexin and calreticulum, and so the protein will get detached from calnexin and calreticulum, and then protein or polypeptide chain will get detached from calnexin or calreticulum, and then it will, there is a sensor, molecular protein that is called UGGT1 or 2. It will detect, it has a capability to to analyze whether the protein has acquired the native structure or not. If the protein has acquired the native structure, it will then pass on to next organelle for further post-translation modification, that is either to ER, Golgi, intermediate, or directly to or sometimes people call it directly to Golgi. And if the protein does not acquire its native structure, then what it will do is it will again add this glucose. Now the monoglucosylated structure will again get attacked, will again get recognized by calnexin or reticulum, and so this cycle will continue. However, this cycle does not goes on for infinity. Otherwise, after some time, there will be a lot of crowd of unfolded protein in the endoplasmic reticulum. In order to take care of this, there is another path that is called ERAD. ERAD. Its full form is endoplasmic reticulum associated degradation. What it does is there is a enzyme that is called ERMAN1. ERMAN1. It is also present inside the endoplasmic reticulum. What it does is when the after a certain cycle, if the protein does not acquire its native form, it will cleave off either of these mannos, either this, or any of these mannos, and now the structure will have only at this moment the Glycan structure will have 9 mannos. However, after this mannos cleaves off this one of the this enzyme ER-1 cleaves off one of the mannos from this Glycan structure. Now this 8 mannos Glycan structure is recognized by the enzyme that is recognized by the protein that one of such example is OS1. Or yeah, I'm not sure about this name, but I will let you know about this name in the next video. Let us just understand the concept that whenever this ER-1 will cleaves off one of these mannos, this structure, which is usually between mannos 5 to mannos 8. Yeah, it can cleave off other mannosidase enzyme can cleave off other mannos also, but this mannos 5 to mannos 8 Glycan structure is recognized by the enzyme that is involved in ERAD mechanism, that is endoplasmic reticulum associated degradation, and that enzyme will helps in transport of this unfolded protein towards the proteasomal degradation through ubiquitination, and we will discuss about this proteasomal degradation pathway in the next video. So that's all for this video. Thank you.